Simple columnar epithelium. We've looked at simple squamous, remember thin and flat cells. To me, they look like fried eggs. Simple cuboidal, areas of secretions. They look like cubes to me. When we put them together as a tube, they look like a pearl necklace. Now we look at simple columnar. They tend to look more like rectangles, but to me, a lot of times I see they look like fingers or stubby fingers. 3 different views of it. So we look at a pictogram of it. One thing I just want to share with you is when we look at more of columnar, the nuclei tend to be closer to the basal surface. Remember we learned our polarity. There's 2 sides, apical surface and basal surface. And basal surface is that deeper foundational layer. And I look at those nuclei and we see them closer to the basal surface. So if I take that picture and expand it down here, I'm going to draw the edge. This is the basal surface here. So going up, this is all my cuboidal cells leading to the apical surface. And you can see all those nuclei that are hugging close to the basement membrane. So to me, I see more like stubby fingers, not squares or pearl necklaces. And then you have a lot more cytoplasm. So you have a lot more room in the cell. Why? The structure creates a function. I have more complex chemical reactions. Where do we see this the most? We see it in our digestive system, the gastrointestinal tract, stomach, small intestine, large intestine. So this view shows it like the lining here. So the food would run along here. If I go to this picture on the right, what this is showing you is the layers of the small intestine. So we're looking at epithelial tissue, and then there's connective tissue, and there's muscle tissue. We'll come back to that when we get to histology of the small intestine. Right now we're just focusing on this ribbon here on the top. And when we see hills and valleys, this gives me surface area. It's one of the key things we'll talk about in the course. The more I have hills and valleys, the more I can put things through. So this is microscopic for that food to drop down in here and start to absorb those nutrients. So when I look at this lining and I just take one of these fingers here, just this one, that's looking at this here. So go back again. This lining is just this lining. So it's wrapping like this, and that's where I can see all these columnar cells individually. And how do I know which ones they are? Because I can see those nuclei. In the middle here, almost like you're looking at a finger, the meat of the finger is connective tissue, whereas the lining of the finger, that's my simple columnar epithelium. So those individual cells are going like this, and so I have a lot of cytoplasm here. I can't see too well the plasma membrane, but I know cell to cell just because of those individual nuclei. So it's wrapping around. When I go to this picture, it looks inside of a tube. Looks pretty close to looking like cuboidal cells, but they are longer, more like stubby fingers. So this is all individual columnar cells, and notice those nuclei are closer to my basal surface. So my basement membrane is that structure, that surface, basal surface. Inside of the tube or lumen, this surface is the apical surface. And I like this picture because it clearly shows nice round nuclei, and then those lining of the cell or boundary of the cell, my basement membrane, my plasma membrane, or cell membrane. So this is simple columnar epithelium.